morning. How are we all doing today? You may have forgotten, I'm still Dr. Wyman. Actually, it's my evil doppelganger. No, still Dr. Wyman. Thank you for coming to our first natural science seminar of this year. We're going to have 5 more of these. Put them on your calendar when they come by. Today, we have the absolute pleasure of bringing back one of you. Any more mature monk? Wow, that's more advanced too. Right. This is Grace. Now, she was Emmert when she was here before. She's sure. She's gotten married. That's what happens. And once she graduated from here in 2015, she went up to Michigan Tech University. She has managed to get out of the snow.
Not yet.
Not yet. Soon, right?
It's coming.
It's coming. She earned her master's degree in biology and is approaching a defense date for her PhD in a few months. We'll be praying for her and her sanity. Also be praying for Charles. Now, let's focus on Charles. Today, she is going to give us an idea of what she's been working on as a PhD student. Now, if you wish to talk to her and find out more about graduate school, biology, career options, coming here and how it ends, she's available for lunch between 11:45 and 1:15 in the DC. And the food is great, right? Yeah, of course. Grace, thank you so much for coming.
Yes, thank you. I'm excited to be here today and tell you all what I've been up to for the last couple of years. So, as Dr. Wyman said, I did my master's at Michigan Tech and then transitioned to a PhD. So I've been working on this project for about 3 years now. And so today I'm going to be talking about upcycling plastic into food. And so just like a quick introduction on who I am and where I've been. We kind of covered it, but I came to Spring Arbor for my bachelor's degree and graduated in 2015. Then I went all the way up to the UP, and we moved up there in 2017. And I've been working on a master's and PhD there. Along the way, I married Charles, and he's on the slide because he's been very important and supporting me through this whole thing, and listening to talk about this at length for a long time. And so now I work in kind of a combination of different fields. I'm mostly a microbiologist, but I also dabble in ecology and bioinformatics, which I'll talk about. And then the project kind of forces me to dabble in chemistry as well as bioprocessing a little bit as well. So I get to explore a lot of different topics, which is amazing. So we'll start off by talking about food. And of course, food is one of our most fundamental needs. We need food to support everything that we do, activities, learning, thinking, just everything is really dependent on having enough food. And unfortunately, food insecurity is a huge problem in our world. Lots of people do not have enough food, especially in countries that are still developing. There's a big shortage of food, and there are a lot of people who are going hungry. About 811 million people are currently hungry in the world, and that's increasing by about 12 million people per year. And so we need creative solutions to address these challenges, or this challenge. The other challenge that I'm going to talk about is plastic pollution. And of course, that's also a big problem worldwide, and I probably don't have to convince most of you that we use a lot of plastic and it accumulates in our environments, and that's not a good thing. And so the very reasons that we like plastic, because it's inexpensive and durable and it lasts a long time and it's cheap, that's what we like. That's what we like about plastic. But it's also those things that lead to the problems that we have. They're so durable, they're hard to get rid of, and they don't biodegrade on their own if we leave them in the environment. And so I have some statistics on the screen just kind of showing how big of a problem plastic is. We are producing a ton of plastic every year, 380 million tons, and we're only recycling, these are statistics for the US, they're a little bit better in Europe. They're actually a lot better in Europe, but we only recycle about 9% of our plastic here in the US, and about 80% of it accumulates in landfills. And so our vision with this project, I'm saying our because this is not just my project, there's a huge team of people working on this project. And so the first part of this is just going to be me giving a big overview of the project, and then I'll kind of dive into what I'm specifically working on. But our vision is to address these 2 challenges simultaneously. So hopefully we can come up with one solution for these 2 problems. And so if you think about the old saying, one man's trash is another man's treasure, that's kind of the basis for what we're doing here. And so Our goal here is to use bacterial communities to degrade the plastic and then harvest those bacterial cells as a source of food that's completely nutritious, it's non-toxic, it promotes health, and it's even personalizable. So it can be adapted for individual needs or the needs of a group of people. And so bacteria, this is where I get to talk about bacteria, I love bacteria, so it's quite exciting to me. So bacteria are very common in the environment. You might think of them as germs, or we talk about COVID a lot, that COVID is a microbe, not a bacteria specifically. But there's a lot of bacteria in the environment, and they do all kinds of different functions that can be helpful, and we can adapt them to use for our purposes as humans. So wastewater treatment is one example of this. We can use bacteria to degrade toxins in wastewater and then repurpose that water. And so many microbes that are naturally occurring can act as catalysts for processes that are relevant to industry and different things that we need to accomplish. So our strategy here is to harness some of these naturally occurring metabolisms and use them to convert waste into food. And so the idea is that we take some isolates of native microbial communities, we feed them plastic waste, we can insert engineered microbes into those communities, and those engineered microbes can be designed to produce certain lipids or fats or other nutrients that might be needed in this food or vitamins, and then we get out this nutritious food out of the process. So if you're thinking microbes, I don't want to eat bacteria, that sounds gross. It's not as crazy as it might first sound at first. So on the screen I have examples of a few different products that are made out of microbes. Most of these are more common in Europe than they are here, but a lot of microbial cells contain the nutrients that we need: proteins, carbohydrates, fats, different vitamins. And the use of microbial cells as food is becoming more common, and it's often used as a nutritional supplement of sorts. And here are some examples, of course. And so this actually has a name, single-cell protein, and I think it should become more common because it's a very sustainable option for food. And then, this is where we get to talk about plastic. So plastic polymers are very high energy cost to break down. So they're very sturdy. That's why if you go bury a milk jug in your backyard, it's not going to just fall apart and degrade and go away, because these polymers are very stable and it's very difficult for them to just biodegrade on their own. So I have a couple examples on the screen of different types of plastic. My research specifically focuses on polyethylene terephthalate, which takes 5 to 10 years under ideal conditions to break down. And of course, the environment is almost never ideal conditions. It's a little colder. It's not the nice, pretty conditions that we have in the lab. And so you notice some of these plastics, like PVC here, we don't know if it ever breaks down because we haven't really been paying attention long enough to know. And so some of these just persist a very long time in the environment. And so we need a creative solution for this because this is not going to work out with how much plastic we're making. And so our plan here is to couple chemical methods with biological methods to break down plastic much more efficiently, so we don't have to wait hundreds of years for our plastic to break down. And so by using chemical methods, we can break our plastic down into more biologically accessible compounds and then speed up the process. So our focus is to design a system where we couple chemical and biological processing to convert plastics into food. So the idea is to build a black box that you can put plastic in one end and get food out the other end. And so this is an overview of the process that we're designing. The idea is to put mixed plastic into the process and go through a chemical conversion step, which I'll talk more about soon. And then we feed those chemical compounds that we're producing to our natural microbial consortia, and then we can harvest those bacterial cells as a food source once they've consumed all of the plastic. And so by coupling the chemical and biological process together, we can speed up this rate quite a bit, and so we're degrading plastic in the order of days instead of decades. And so this is another view of our system. As I said, we're putting in mixed plastic, so we have kind of two paths feeding into our microbial breakdown step. So the first one, we're looking at high density and low density polyethylene and polypropylene. And we're breaking those down with a process called pyrolysis, which is essentially just very high heat, and that produces alkenes of different sizes. And then where my work kind of comes in a little bit more is with ester-based plastics such as PET, polyethylene terephthalate, and that goes through a different type of chemical deconstruction, which we'll talk more about during this talk, and that produces these aromatic monomers and ethylene glycol. And then both of these can be fed into a microbial breakdown step and then recovered as biomass. And so we'll talk about pyrolysis first. I'm not going to spend a lot of time talking about pyrolysis because it's not the focus of my work. But essentially pyrolysis is where we heat up plastics and then they turn into alkenes. And these types of plastics, you might know them as like grocery bags, milk jugs, that sort of thing. And so this kind of looks like a hot mess, but this tank right here is the melting tank. So this gets really hot. We put our solid plastics in here and they melt, and then they get pumped through this pipe here, which it's probably hard to see in the picture, but we have some heating elements wrapped around there. And it gets up to about 600 degrees Celsius here. And so it gets pumped through there, and then the products are collected from this end. And out of this process, we get three types of products. We get a solid, a liquid, and a gas. And the gas product can be used for power generation. I'm not really sure how that works, but the team that's working on this does. The liquid product can be fed to our bacterial communities. It basically looks like an oil. And then the wax product can be sent through the reactor again to produce more of the gas and the liquid. And so this is a gas chromatograph of the liquid product, and I'm just showing you this to demonstrate how many different sizes of things we have. And so that's one benefit of using a microbial community, because we can deal with these complex mixtures of things, because we have a lot more metabolic potential in a community compared to an isolate of one organism only. And so now we're going to transition to talking about the ester-based plastics, or the PET. And so this goes through a slightly different process. We still have a lot of heat in this process, but we're also adding a chemical step. And so by taking our PET polymer and reacting it with ammonium and hydroxide, we have two different chemical processes happening at the same time. We have hydrolysis and ammonolysis happening. And so we're producing three possible different monomers depending on which of these reactions is happening more than the other. And so the one that is mostly produced is terephthalic acid, which has these two carboxylic acid functional groups on either side. And then for each of these aromatics that's produced, we also get a molecule of ethylene glycol. We can get terephthalamide, which has an amide functional group on either side of this aromatic ring. Or we can get the terephthalic monoamide, which is one carboxylic acid and one amide functional group. And so our deconstructed PET is a mixture of all of these components. So ethylene glycol and then the three aromatic molecules. And so by using this process, we can solubilize 90% of post-consumer PET, which is pretty exciting. And so you can see in this picture, this is what our liquid looks like. This is actually water soluble, so it's not an oil. It'll actually dissolve into the water, which makes it a lot easier to work with. And then over here, you can see the solids that were not solubilized. And so here, this is just to give you an idea of how we measure bacterial growth. And so we do that using optical density. So if you look at this picture here, we have two flasks. This flask has our media in it, and this flask is after it grew. So this flask has cells in it. So we can use optical density to measure the cell's growth. And so the way we do that is we just measure optical density over time. So that's what these plots show. They show the cells increasing in density over the course of hours. And these communities we got from different locations that we have samples that we've gone to sample, or that we have collaborators in. So Hancock is about 10 minutes away from the university, and so we have one. This is from a compost from a farm over in Hancock. This community came from a stream in Pennsylvania where we have some collaborators. And then Michigami and St. Ignace are other places in the UP where we collected samples and got these different communities from. And so that shows you that these metabolisms to degrade these compounds are extremely widespread and common. And so what we do is we take a scoop of dirt from a location and we kind of force feed them these plastic breakdown products, and we dilute out the organisms that can't survive. It's kind of survival of the fittest, and we end up with these communities that are able to degrade these compounds. And so just kind of the process again, now that you know what the chemical deconstruction steps are, we have our different types of plastic being converted into either alkenes or these aromatic molecules, and then we feed them into the microbial breakdown step and collect biomass at the end. And so why use microbial communities? I talked about this a little bit, but the benefits of microbial communities are that microbes naturally in the environment live in communities. It's very rare to see a microbe living alone somewhere. It's often an artificial thing that somebody tried very hard to do. Interactions within the community can lead to more efficient processing of inputs, so they often have interactions with each other and work together and share metabolites to support growth. And a diverse community allows for more flexibility in dealing with changing environments and changing inputs. So you can kind of think about it like a group of people, like all the people in this room here have different skills and talents, and if we take all of us together, we could probably do more than any one of us could do alone. So bacteria are kind of like that as well. I like to think about them as people because I spend a lot of time with them. So if we think about, I'm going to introduce two concepts here that are important for my dissertation work, which I'm going to dive into in just a minute. So if we think about the degradation of terephthalate, which is this molecule, we can think there are a couple different ways it could be broken down. We could have just one organism doing all the work, and it has the entire metabolic pathway that it needs to break down the terephthalate, or we might have one organism doing the first step and passing those metabolites to the second organism that does another step, and so on. So we might either have teamwork or we might just have one working by itself. And so we call this kind of top scenario where they pass metabolites back and forth, division of labor. So they're working together to accomplish a task. And then this would be just one organism working alone, so that would not be division of labor. If we think about this community processing two different inputs, we can think about either specialists or generalists. So a generalist would be this organism right here. It can process two different types of inputs, whereas this organism and this organism are specialists, and they only really like one substrate in this example. Now, of course, real life is a bit more complicated than this, but a lot of my dissertation is looking at division of labor and then specialists and generalists within those communities. And so getting into my actual dissertation now, I have three questions here that I've been working on. And so the first one, we want to know, can our natural microbial communities actually degrade this chemically deconstructed PET? Because it's been shown before that we can make this chemically deconstructed PET, but can the bacteria eat it? The second question, is there division of labor in the communities? And then third, are there specialists and generalists for the different chemical components of the deconstructed PET? And so we do know that naturally occurring bacteria can degrade PET. However, it's extremely slow, as I mentioned before, and the rate limiting step is this depolymerization with this enzyme called PETase. But the rest of this pathway can happen very quickly. And so organisms that can degrade PET have been isolated from many different environments, marine soils, sewage, and we've even identified this organism, Ideonella sakiensis, who can produce PETases and depolymerize PET by hydrolyzing the ester bond, but that's still very slow. And then the microbial degradation of TPA, or terephthalic acid, is much, much quicker. It can happen in as little as 19 hours. And so digging into the first question now, I'll start to show you some real data. So just a quick refresher on making the deconstructed PET. We have our PET polymer. We put it through these ammonolysis and hydrolysis reactions, and we get this mixture of compounds. So we get the terephthalic acid with the 2-carboxylic acid molecules here, the terephthalamide with the amide functional groups, and then we get the compound that has one of each. And then we have ethylene glycol. And so if we look at this process, we can take the PET, and in this step we're adding nitrogen. And then if we neutralize that, because this would be a strong base after we deconstruct it, before we feed it to our bacteria, we have to neutralize it to get a neutral pH, otherwise our bugs aren't going to like that very much. And so if we do that with phosphoric acid, then we have this feedstock that has nitrogen and phosphorus already in it. And that becomes much more efficient. So there's a question of, do we need to use our fancy microbiology media at that point? Because we've already added 2 of the main nutrients that they're going to need. And so I did an experiment where I grew, we like to name our communities after people in the lab. So this is Emma's community and this is my community. It's just kind of more fun that way. And so in this blue box here, all of these communities were fed deconstructed PET. And this community, we fed them just terephthalic acid as kind of a positive control. And then we grew them in 3 different conditions. So we had sterile millicue water, no added nutrients. We had sterile river water, which I filtered and autoclaved to kill anything that's in there, because we wanted to see if we could scavenge water from our surrounding environment. And then we also have our fancy microbiology media. And if you look at these are the same type of growth curves that I showed you before, where we have optical density here and time on the x-axis. And you can see there's not really any difference between how they grow on the model compound and how they grow between these treatments, which is really exciting because that means that the nitrogen and phosphorus in our PET is supporting the growth of our organisms without fancy media added. So that makes this process just that much more sustainable, which is really cool. And I did do some statistics on this, and there were no statistically significant differences between any of these, which was super exciting. And so, but that shows that they grow, are they degrading it? And so here I have HPLC data, or high performance liquid chromatography data, showing that our starting compounds, which this is our PET over here, and then this is our terephthalic acid, our model compound over here. And we show quite a decrease. It looks like the Laura community ate more of the PET than the Emma community, but we degraded quite a bit of it. And then if you look at our model compound, they degraded almost all of it, which is incredibly cool as well. And so we're still looking into what exactly the Emma community is doing. That will probably be an ongoing question throughout this presentation, because they're different communities from different environments, enriched in different ways, and so there are still some open questions as to what makes them different. And so after we saw that they could degrade the plastics, we wanted to know who's there. What are the names of these organisms? Have they been seen before? That sort of thing. So what we did was 16S rRNA sequencing. And so this is a quick way of sequencing a community. And so we can take our bacterial chromosomes and we can take out just one gene. Using PCR, we'll amplify the DNA to get only that gene. And then we will sequence just those little chunks of DNA and we'll get names for the organisms. We don't get to know all of their potential and what exactly they're doing. We'll get into that later. But right now we just want to get names. So this is kind of like fingerprinting. If you think about that, you can kind of tell what somebody's name is, but you don't necessarily know what their occupation is from their fingerprint. So these are our microbial communities. So we have our communities from Emma 2 and Laura 1, our enriched communities. And the first thing you probably notice about these bar charts is that there's a lot of orange. And so that orange represents an organism called Rhodococcus. And that organism in all the sequencing we've done has been very dominant in our communities. Some of the other important organisms here is this blue color, and that's Hydrogenophaga, which is another known to degrade different aromatic compounds and different toxins in the environment. I have a list at the bottom of the screen of all the different compounds I could find that this list of organisms here has been involved in degrading. So all of these genera that you see listed here have been shown to degrade all kinds of different compounds and contaminants. Other important organisms that you'll hear more about later is this Schaerer organism that's in red at the bottom here. I believe that is an ethylene glycol degrader, but I'll get into why I think that later. So these are what our communities look like. Our Emma community is a lot less diverse overall. It's dominated pretty much by Rhodococcus, and then the Laura community has a couple more different organisms in it. So in response to the first question, can microbial communities degrade chemically deconstructed PET? The answer is yes, they can degrade it, and we kind of know who's there, who might be doing the degrading now. So the second thing I want to know is we have some names, but how do these organisms work together or compete to degrade the PET? And so there's a couple of different scenarios that could be going on. We might have division of labor where they're just happily all working together and sharing metabolites. Like this guy does step 1 and passes something along to this guy, who does step 2, and they're all just completely happy doing their own task. Another scenario is that we have one organism that's doing all the degrading, and we have other organisms producing different nutrients that the main degrader needs and degrading contaminants that might be building up in the environment. And so we have one degrader and many different helpers. Or they could all just be fighting with each other and competing for the same substrates. And so we're going to try to untangle what exactly is going on in the community. So to do this, we're going to do what's called metagenomic sequencing. And so again, we're going to start with our bacterial DNA. But instead of just taking out one gene to look at, we want to look at all of it. And so what we do is we break up our bacterial chromosomes into short sequences, and we sequence them, and then we try to put them back into genomes. And so this is kind of like taking a book and putting it through a paper shredder and then trying to tape it all back together. And so it's not a perfect process, but you can get a pretty good idea of what each organism is doing. And this tells us what they have the potential to do. And so it doesn't necessarily tell us what they're doing, but it'll tell us what can they do. And so looking at this, I know this graphic is a lot, and in the next couple slides I'm going to break it down and kind of point out the important things that are going on. But right here, this is just to get the big picture. So I have a couple different communities here. We have the Emma community, which you already met. We have two Laura communities. We have a Lindsay community and a Steve community. And so we have each of these different colors represents different pathways that are needed to break down these components that we're feeding them. And so in red, we have the alkane degrading pathway. We have the catechol pathway in orange, the ethylene glycol pathway in green, protocatechate in blue, and terephthalate in purple. And the important thing to notice here is the color blocks. So if you look at the purple and then kind of try to follow it across the screen, you can see that the organism that has the purple doesn't have the blue. And so that points to division of labor. But because this is difficult to look at, we're just going to focus on the LoRa 2 community for now. And so we'll just look at that, and it will hopefully make a little more sense as we move on. So looking at our 16S rRNA sequencing again, we can see the proportion of different organisms in that community. So you can see we have a lot of Rhodococcus, a lot of Hydrogenophaga, and then we also have in green this Pelagibacterium, and in dark blue are Shalatococcus. And we have 4 high-quality genomes that we got from our metagenomic sequencing. And so if we think about our enzymes as different shapes, some of them have multiple subunits that work together, some of them just are one piece, and we look at which important enzymes are produced by each of these organisms. And then we think about our metabolic pathway. We can kind of throw the shapes on there as catalysts, and we can kind of start to see how this pathway plays out. And so just looking at these 4 organisms, of course there's a lot more going on in the community, but these are the 4 that we have good sequencing data for. We can see that the Hydrogenophaga seems to kind of have a monopoly on these first 2 steps. It's the only one that seems to be able to do those steps. And that can explain why it's so abundant in our communities. And then we kind of, when we look at the next step, we can see that the 3 other organisms all compete. It looks like they produce the same enzymes to do that step. So we have 3 organisms kind of trying to do the same thing. However, if we look up a little bit, we can see that the catechol pathway is an alternate pathway that we can go through instead of the protocatechate pathway. And of course, our friend Rhodococcus produces the catechol pathway. And so that could explain why we have so much Rhodococcus in our community, because it's taking a little shortcut and getting around that competition. So here, this nicely shows that we have some division of labor. We have the Hydrogenophaga that is only able to do one part. And then we can also see that there's a little bit of competition going on as well. And so, is there division of labor in these communities? So far, our data points to there being division of labor and the different organisms working together. So the last thing that I want to look at today is, are there specialists and generalists for the different chemicals in the deconstructed PET? And so to do that, we're going to take a little review about DNA to RNA to protein. And so we know that when cells make protein, they have their DNA, which is transcribed into RNA, which is then translated into amino acids, which are folded into proteins. And so if we want to see what the cells are actually, which proteins the cells are actually making right now, we can look at their RNA transcripts to get an idea of what are they doing right now. And so we kind of do the same thing. We already have our metagenomic sequencing, and so we can sequence our RNA transcripts and then compare those to our metagenomes to see what are they doing right now. And so one analogy for this is if you think about the lights in this room, the metagenomic sequencing will tell us which lights we have available, but the transcriptomic sequencing will tell us which lights are currently on. And so we're trying to look at the current, like right now, activity of these cells. And so we did that, and so I'm just going to focus on the LoRa1 enrichment for right now because that's the one that we have transcriptomic data for. And we can see the colors in this graphic don't really matter so much, but we can see the expression of different genes. So all of the different colors represent a different enzyme that's involved in breakdown somehow or another. And we can look at the overall expression when we grow them on different compounds. So in this first column, we have the deconstructed PET. Second column, we have ethylene glycol. And then here we have the amide compound. And then over here we have terephthalic acid, the carboxylic acid compound. So we can look at the different organisms, and I'm just going to focus on the few that we've been talking about already. And what we're looking for is specialists and generalists. So we're looking for organisms that either grow on all of these or only one of these. And so if we look at our rhodococcus, we can see it is actually pretty active in all of these. It's a little less active on the ethylene glycol, but it seems to really like both of the aromatic compounds, which is pretty fun. And then we have our hydrogen omega up here, and that does really well on the mixture of everything. It does okay on the ethylene glycol, and then on the amide compound, it does nothing. It does not seem to like that amide compound at all. And then on the terephthalic acid, it does pretty well again. And so that's an example of some preference there. It's not exactly a specialist because it grows on the ethylene glycol and the terephthalic acid, but it does have some preference. And then we'll just also point out this Schaerer organism that I mentioned kind of at the beginning. It doesn't really grow very well in the mixture, but it does great on the ethylene glycol, and then it doesn't really like the aromatic compounds very much. And so that one might just be an ethylene glycol specialist. And of course you can look at this for a long time and just really kind of guess how they might be working together, which is pretty fun, but I'm not going to go through all of it right now. But it's very exciting to see that we do have some preference on the substrates. We seem to have some specialists, some generalists, and we're still working on getting better sequencing data to really flesh out this picture a little more. And so kind of in summary of these points are the 3 points I talked about. We've shown that the microbial communities can degrade our chemically deconstructed PET. We've shown that our genome sequencing suggests that there's division of labor within these communities. And we've shown that the gene expression changes when the microbes are grown on the different chemical components of the deconstructed PET, showing that we have specialists and generalists. And so just our process again, looking at, we've talked about the chemical breakdown steps, we've talked about the microbial breakdown step, and now we'll talk really briefly about the biomass recovery step. And so this is still very much a work in progress, but we're moving towards getting a safe single-cell protein product at the end of this process. And so what we're doing to make sure that this is safe, we're doing chemical analyses to look at different toxins and their concentrations. So we're doing metabolite extractions to make sure that our communities aren't producing any toxins. We're doing whole animal screening for toxicity. So right now that means we're feeding the biomass to worms and looking at the development of those worms. And then we're also just looking at our metagenomes and kind of really digging through. That's a ton of information, and looking for any genes that might be producing toxins there as well. And at some point we're probably going to have to do rat or mouse studies to make sure this is safe as we move forward. And then this is just a picture showing the final products from our process. And so I meant to borrow this from my advisor to pass around, but I forgot to do that, so we'll just have to make do with the picture. So here's our raw PET that goes into the process. This is what it looks like after it's been deconstructed and neutralized. This is our HDPE. And then we have our pyrolysis products. So we have the wax here, and then the oil here, and then this is what our final ground dry cells look like at the end. They're actually bright orange, these communities, when we grow them, so that's why you see this like kind of weird color. They look a little bit like brown sugar or orange juice when they're actively growing, which is kind of gross, but I think it's kind of cool. So my conclusions, we've talked about how coupling chemical and biological processing of plastic improves the efficiency. These methods include heat and chemical methods to convert our plastics into more biodegradable compounds. Our microbial metabolisms that use our plastic as carbon are widespread. We've found them in many different environments across the UP and other places that we have collaborators. We've found generalists and specialists within our communities that are cooperating and competing to metabolize our plastics. Our microbial communities are converting our deconstructed plastics into biomass, and then we're doing further work to see if the biomass is going to be safe and nutritious to eat. And so with that, I'd like to thank the huge team of people that's working on this project, in particular Dr. Steve Techman, my advisor. This whole project was his idea, and he's kind of mediating all of these different teams of people. We have chemical engineers, we have people at the Great Lakes Research Center, we have people at the University of Illinois and Western University who are also working on this project, and so it's a huge team of people who are all working on it together. It's been really fun to work on. So with that, I can take any questions that you might have, and then my email address is on the screen if anybody wants to email me later on other questions. Go ahead.
Some of those organisms you had are organisms that would prefer to attach. Have you tried seeing if surfaces increase cycling of the molecules?
Yes, so because we're trying to build a system, we actually don't want biofilm formation. And so we're actually trying to avoid attachment to surfaces. So
That'll increase your diversity.
but because we have to pipe them through this, like we have to pump them through tubes and stuff, they're worried about clogs in our system.
If you're aerating the system, putting a surface in them, that's not hard.
Yeah, it seems like they're pretty happy and they don't like to attach. When I grow them like in a flask in batch culture, they are all over the sides and we get these really thick, slimy chunks. But once they're in the bioreactor, which I haven't actually seen yet because our chemical engineers are working on the bioreactors, but I've heard that they don't attach as much. I think it's because they have more oxygen and they're not limited by oxygen.
Well, that was my second question.
Yeah.
Do you know anything about how oxygen tensions, how oxygen concentrations affect your degradation of your plastic?
I am guessing they affect it a lot because we have grown these on stir plates where they're just slowly stirring and they don't get as aerated and they don't grow as fast. But then if we put them in the shaking incubator where the plastic is being shaken around, we get a lot more aeration and they grow a lot better. And so that's very anecdotal. We're just now getting them into bioreactors actually this week. So they're probably doing it right now actually. But yeah, so we're very excited to see how fast they'll grow once we get them in. Because if you look at like our growth curves, when they're growing fast, like they hit log phase and they grow really, really fast and then they level off. And so hopefully in the bioreactors we can really fine tune the conditions to keep them in that exponential growth phase and produce
Have
more
you tried
biomass.
chemosteps?
I don't think so.
Which is another reason why you wouldn't want surfaces, because I
Yeah.
call it chemostep.
Yeah.
Other questions? Go for it, Tom.
Which slide up there that you showed today represents the most amount of time you spent in the lab, and how much time was that? If you just show whichever one it was.
That's a good question.
Or you can just pick one. It doesn't need to be the most. Like your
Yeah.
growth curve.
Yeah, the growth curves are always a lot of work because they're always going in either every day or multiple times in a day and taking measurements. The most time-consuming experiment that I actually did this summer, and the data is not quite ready for a presentation yet. So that would be my most. Yeah, and then a lot of this is computational as well. Like this was a ton of computational work, so just hours and hours of writing code on my computer. But the lab side of things was actually pretty easy for this one. But then I would say the growth curves have to be the most time intensive as far as lab time.
So that growth curve that you showed, that whole slide,
Yeah.
would be months maybe?
That experiment was about a week. I just finished an experiment that was a whole month of every day of 180 some tubes. So that was my biggest experiment so far. It was kind of like my grand finale for my thesis. But that's not ready for public viewing yet. It will be soon.
Go ahead,
Go
Garnet.
ahead.
What advice do you have to this group of students here about what they should do now to prepare for this kind of work, not necessarily in biology, but in any graduate program?
Yeah, I would have to say get research experience if you can. A lot of people either don't, like I didn't have a lot of research experience going into it, and so I was very unsure if I would like it, so I kind of started out really cautiously. I volunteered in the lab that I work in now, and I was like, well, it's kind of fun, so I'll do a master's, and then by then I was really entrenched in it, and I was like, okay, I'm going to do a PhD. But if you can go into it knowing that you enjoy research and that you like that, that would really help you out in the long run, I think. And then the other thing is just find a project that you're really passionate about. You really need to enjoy it. Otherwise, when you're doing 180 something tubes every single day, you're going to really not have fun. So you have to really care, or just be somebody who cares a lot about everything, and then
And
you're
it helps to
good.
be in a lab with nice people.
Yes,
Yes.
yes. Nice people are extremely important.
Yeah.
I
A
have
lab
friends.
full of that kinds of horses can make your life pretty easy.
Yes, I'm very lucky. My advisor is wonderful to work with, but I have friends who are not so lucky, and they have had a very miserable time in grad school. So finding a nice person to work with makes a huge, huge difference.
Well, before we thank Grace again, I want to point out a couple things. One, this young woman didn't say much over 4 years.
Yeah.
That was awesome. That was flat out awesome. Great. Well done.
That's
Well
nice.
done. Two, please notice something. She only showed one biology slide.
That's right.
She showed math,
Yeah.
computational biology, computational science. She showed organic chemistry. I thought Baldwin here was going to have like shock, like on the floor. I'm so excited. I'm glad to hear him. Like, you're trying to do this. He's just vibrating with joy, like electrons. She did chemical synthesis reactions. She did metabolic reactions. She did metagenomics. She did non-genomics. She didn't even talk about the molecular biology she carried out because she didn't have time for such things. When you engage in science, you don't do one thing. So as much as I enjoy listening to you all come out and tell me how much you dislike X, Y, and Z, you know what I don't like? Liver and cauliflower and broccoli. And yet I eat those things because I'm old. I'm getting older. Life isn't about what you like. Life about what you need to do. We all do it. She's doing it. Do not look at a topic and go, oh, I don't like that. Yeah, you don't have to like it, that's fine. Lean into it anyway and do the best you can to command it. Because if you can command it, you can use it. You can use it for his purpose. Grace, well done. Well done.